Hi everyone, I'm Sharzad and I want to talk to you guys about how I collect soil samples for microbial analysis. For this experiment, I'm looking for bacteria which are mostly anaerobic, so I'm looking for the parts of the soil which are saturated. So with this tape, we're trying to measure and find the water table to find out where the actual saturated zone in the groundwater is.
5.66 feet.
So now that we know where the saturated zone is, we are going to collect the soil samples. So we are using the judgmental sampling method, which means that based on our previous information and data, we have selected a few different locations in the site to collect soil samples. We are using direct push method using a geoprobe. So this method is used for different purposes for investigation of groundwater and soil samples, and the drilling equipment is used to push a hollow steel rod lined with a plastic sleeve into the ground to a targeted depth. The soil sample will be collected in the plastic sleeve, and the plastic sleeve later on will be pulled up again, and then we can study the soil that is collected in the sleeve. It is very useful for studying the vertical geology of soil, or in my case, I want to study the microbial community at different depths of the saturated zone of the soil. So now you can see the plastic sleeve that I talked about with the soil sample in it. So we collected a soil sample from 8 to 12 feet below the ground surface from this specific location. And now you can just see how the geoprobe pulls up the hollow steel rods, and then we're ready to move on to the next location. So we continue the process until we collect all the soil samples from the desired depths from different locations that we have specified. So here I just want to show you guys how I prepare the samples before shipping them to the lab. So I cut them usually in 1 feet sections, um, as you can see. And then I usually use this plastic wrap to wrap both ends of the sleeve, the plastic sleeve, to prevent moisture loss and minimize the interactions between the soil and air as much as possible. And after wrapping the sample in the plastic wrap, I'm gonna put them on ice in the cooler to be shipped to the lab. I also make sure that I label everything very accurately. So the samples were shipped on ice to the lab around 4 degrees Celsius. And I usually try to process them the same day before putting them away for long term storage. In this case, I'm collecting some of these soil samples for DNA or RNA extraction and later on analysis. So at first, I have to try to homogenize that 1 foot sample that I have. So as you can see, I use a clean plastic bag to um, collect that 1 foot soil sample, and then I'm gonna mix the soil sample inside that plastic bag to make sure it is homogenized. So later on, when I'm taking the sample out of the freezer, I'm pretty sure that that sample that I'm taking for DNA extraction is a representative of the whole 1 foot that I had. So now the soil sample is homogenized as much as possible, and now I'm just collecting the soil into 50 milliliter vacuum tubes, and then I'm gonna close it and wrap it in plastic wraps again, and then the sample is ready to be stored in -20 or -80 freezer for a much longer period of time before DNA extraction or RNA extraction. I like to use some of my soil samples for an activity test. So I'm not gonna put them in the freezer. I'm gonna actually try to keep them alive, and later on feed them with the electron acceptor and an electron donor that they like, and find out if they're able to degrade the contaminant that we are targeting or not. So the bacteria that I'm looking for, as I mentioned before, are anaerobic. So I have to try to minimize oxygen exposure. And for this purpose, I'm doing the rest of the preparation for these samples inside an anaerobic chamber. So, an anaerobic chamber, as you can see, is this big vinyl chamber that is tightly sealed. We have filled it with a gas mix containing nitrogen, CO2, and hydrogen with zero oxygen. So, it is being constantly monitored that there's no oxygen in here. And I do the same thing. I try to collect the soil sample in a clean plastic bag and mix it and homogenize it to prepare it for storage. And later on, when I collect the soil sample in a clean Falcon tube, it can be stored inside a refrigerator in the anaerobic chamber, or it can be stored in another tightly sealed vial and be transferred and outside the anaerobic chamber.